apparently because
she's a super strong.
I'm basically good at transition. Same thing with you guys are officially joining the department.
She's always there. They always have our
I think it should be fine.
Yeah, I was having issues because like I went fine doing staff masters.
Oh yeah.
Like, yeah.
I thought about that and then I'm like maybe not.
When many different departments have their different rules.
I have some of my favorite heroes.
She can't declare about mathematics until the 16th of the 5th, and she has to submit to statistics for the 15th. And the department that she is in has to sign off on it. But to me, I found that they will accept her application. Know that my signature will be valid until the 16th.
Doing your application for 24 hours is fine.
It's going to change things. Instead of like 30-meter range.
I thought about doing the staff masters, but I think in the end
But it's going to be twice as high.
it's not worth it to have the class requirements. Like if I want to learn it, then I just
And then he'll get a speed boost by wall banging.
I heard it's tough.
So that's something I
After that, Zena has to Stats, DSTP. Yeah, I mean, you will, if you get into the cancer proteomics, you will have to take some courses.
where I can watch people like DPS stacking.
Yeah.
That's like the best was horrible. He's just like climbing the skyscraper in the middle of like the clock tower in Eichenwald. He just spirals up. I don't understand how he does it because I've tried to do it a million times.
I talked with Phil, who used to be on of that one.
But the things that he'll be like wall riding on, I'll try to do later, and it's like I gotta get like stuck on a bumper or something like that.
He rarely got
The answer.
All right, welcome everyone. Tools and Technology Seminar Series. A sign-in sheet went around. Hopefully you all signed it. So I'm actually going to let Peter Ferdolino introduce our speaker today.
Thanks. So speaking today is Rucheng Diao. She is a graduate student in my lab. So Rucheng got her bachelor's from Sun Yat-sen University in China and had done undergraduate research both on the wet lab side of things, working with some cell lines, and also had some bioinformatics experience. I met her at one of the bioinformatics kickoff events where she saw one of my talks and asked about doing a rotation in my lab, sat down, started doing fantastic work right away. She was a master's student at the time and recently transitioned into the bioinformatics PhD program here. So we're really happy to have her in the lab, and she's going to be talking today about some work she's been doing on finding some interesting regulatory connections in E. coli by mining high-throughput sequencing data and microarray data as well.
Do I need this?
You don't need that.
Yep. Thank you, Peter, and thank you, Marcy. Yep. Thank you, everybody, for coming to my talk today. So I'm going to talk to you guys about my lab project that I have been working on. It's about like identifying regulatory logic in a curated collection of E. coli expression data set. So I would like to start with introducing something about E. coli. And E. coli essentially have like about 4,300 genes in the genome. They are all well categorized and have a very good established numbering system of it. And unlike many systems you guys may have been working with, 90% of the E. coli genome is actually coding. So there's there's very little waste in their genome. And also it has no nucleus, no splicing, and they have short doubling time and very large population can happen in a very short time. So also very interestingly is that they basically live and die by the transcriptional regulation because everything happens so fast. So especially given there's no splicing and very limited post-translational regulation going on. So it's a very interesting phrase that I would like to cite, that they live and die by the transcriptional regulation. So that's so much about E. coli. I would like to start with an example of how cells like E. coli can get prepared for environmental changes based on current cues. So here we are seeing two genes here. BO3 is a gene that utilizes high oxygen environment, and BD is one utilizing very low oxygen environment. So when oxygen going down, we can expect that the one utilizing high oxygen environment is going down, which is VO3, and the one is using low oxygen environment would be going up. That's kind of what we expected. But if we are not treating it with like downshift of oxygen, but instead with an upshift of temperature, we can still see very similar, like highly correlated pattern happening, that the one that is dealing with high oxygen environment is going down, and the one dealing with low oxygen environment is going up, as if the cell is preparing it for like a low oxygen environment. In E. coli's case, this makes sense because when the temperature goes up, it usually means that you just got swallowed by someone. So you are going to be hit by biosalt, low pH, low oxygen, and all kind of adverse environment stresses. So this is like what E. coli makes sense out of the structured environment that it has been living in. So the takeaway from this slide is like it's regulating more than it needs to deal with the current stress, just like it's regulating the genes that's related with oxygen when it is hit by actually a temperature change. And here we would like to extend this example a little bit. If we put all the environment events on a timeline, as the green boxes here, and we can have an environment event. For example, in the previous example, that would be going down of the oxygen. And then we have some cues happening ahead of the events on the same timeline. And in the previous example, we can find an example as temperature going up as the cue. So the classic view of how E. coli responds to it is a homeostatic way. So that means that it responds when it actually got hit by the stress itself. If we project the environment as the red dash boxes here, we can see there's a little lag here. And there's a new framework for E. coli's gene regulation. It's the anticipator regulation that suggests that E. coli actually start regulating and prepare for the environmental changes once it gets hit by the cue instead of the environment. So if we project it here, we can see the lag is slightly better. And E. coli is actually preparing for this kind of environmental changes. Notice here that given that cue and environment are probably highly correlated, but they are not perfectly like predicting the environment change, so there will be chances that an anticipatory way of responding is actually not the optimal. And here we want to ask the question is that, can we identify and predict stress pairs that will show anticipatory behavior using mostly computational work in my project? So I will start with a fitness data set that is from the cell resource paper from Nicholas, etc. And they basically, that is how their experiment is set up. There is a clear mutant collection library of the E. coli, and they're basically like finding these knockout mutants in E. coli and plate it using robots onto the plate and grow it under certain stress environment and see how big it grows, and record the colony size and score it quantitatively using kind of a modified z-score into a fitness score. And that will give us a heat map of mutants versus stress on their fitness scores. Nicholas do the stress of like 324 different environmental and chemical stress, including a lot of like environmental changes, chemicals, and a lot of them is antibiotics. And one example of the data would look like this at the bottom. Is if we take 45 degrees Celsius as the example, we'll see a score, a fitness score for a deletion of the gene. So we might think about like if we can actually think about if a gene is helpful or hurtful using this data, because when let's go back to our like previous example. If a gene is helpful, we can predict, for example, the high oxygen, the low oxygen gene would be very helpful in this low oxygen kind of environment. And if we plate it on the plate and hit it with low oxygen stress, we can predict that the one knockout with the low oxygen dueling gene would be going worse, and the one knockout with a high oxygen dueling gene would be going better or at least neutral. So we can find out that the helpful genes are actually those with a negative fitness score, because knocking out the helpful gene will make it doing worse in the stress environment. Everybody clear on that? Yep. And besides this fitness data set we find as a resource, we can set out on GO, which is gene expression anonymous, to see if there's any other gene expression data set that's publicly available. And what we want is like vanilla plain MG1655 or similar baseline strains of E. coli with no genetic modification, and is dealt with stress or drug challenges. What we find there is that there are about 780 E. coli data set on GO, and for out of the most recent 400 of them, 43 of them is falling into our category. I will be very interesting to see what happened to the rest of 300. I'm expecting maybe slightly more than that, given that this kind of study is done a bit earlier. So out of these data sets that we can see, half of them are done in 2-color microarray, and about over a quarter is done in 1-color microarray, less than 1/4 is done in RNA-seq. And one example would be, like how the data would look like after the analyze would kind of look like this, that we have a stress condition, and we have the list of the genes, and we have the log to fold change of them. And in analyzing the metadata of the curated data we have on hand, we find that it has the collection has a high diversity in both the biological and the technical side of it. Biological speaking, of course, there is a diversity in the category that people want to treat E. coli with. Well, they actually treated one data set has a microgravity like stress, so there is some thinking. Yep.
So just one thing I think it's worth noting also that each data set that you were showing on the previous slide, there are often more than one stress condition.
Yes.
Set, right? So you got a bunch of different conditions.
Yep. So there is like one data set that has several samples in it that can have several different conditions in it. For example, one data set might study 4 different antibiotics and put them in one data set, but they're in like 4 different samples.
Are the controls for the different data sets the same?
Yes. Most of the data sets come with their own control. For 2-color microarray, they usually have their control as one of the colors. For 1-color microarray, they usually have an untreated sample as in their data set. Does that make sense?
Yeah, but I mean in terms of
Yeah.
media and in terms of the conditions,
Yeah.
are they usually the same across data sets?
That is something that we want to study in the metadata. And for example, the time point when they collect the data, the OD value or like log phase or stationary phase varies. And I actually have the idea of analyzing it with log phase and stationary data and see how much it changes. And like, for example, if you're not doing heat shock or cold shock, most of them are collected 37 degrees. And that is something that is in consensus, and like media sometimes changes, but most of the time you can find good documentation in the paper to find out the details about that. Yep. And like many of them are environment stress, such as heat and cold shock, oxygen, and maybe microgravity. And many of them also study the antibiotics effects. And
So they are all the same. They are not like different strains. Like I'm
No.
used to, looking at genetic diversity in humans. I mean, I assume there is genetic diversity in E. coli as well.
Yep. So the 2 strains we are looking for is MG1655 and BW2. Yeah. And those are the 2 strains they use in the knockdown mutant that is like similar enough, and it's like the baseline of the studies. And technically speaking, yep, we have a very colorful pie chart here, because people use different platforms to do their microarray data processing and RNA-seq data processing. We can see a huge chunk of it is coming from Affymetrix, and the second winner is a Agilent platform. But otherwise, many of them use their own platform, decoring their universities and things like that. So many of them has like only one data set that's coming from one specific platform. So that adds to the difficulty of analyzing and normalizing the data. So how do we connect the resource data together now that we have a resource paper that has finished data and a lot of like gene expression data sets from GEO, and we can use gene expression as a bridge. So a way to think about it is that in the finished data, we identify helpful and hurtful genes. And in the GEO expression data set, we can find upregulated and downregulated genes. So that gives us 4 combinations. And if you're upregulating helpful genes or downregulating hurtful genes, that will give us an anticipatory kind of behavior, just like Murphy's jumping over and land pretty well because he already knows what to do. And if you're downregulating helpful genes and upregulating hurtful genes, you're actually sensitizing the cell because it will probably do worse in the upcoming finished conditions. Just like Neo doing that for the first time, epically failed. So it's very easy to think about it in the first place because we can find a gene 1 and accordingly in the finished data set, delta gene 1's finished score. So that is for one gene there. And we are trying to do it for multiple genes, like 4,000 of them. And we define something as anticipation index. That is, you can think about it as a way of correlation. There are multiple ways of doing it, like mutual information, Spearman correlation, and we also define our own delta score and LFC score. And for mutual information, we're basically looking at the inherent dependence of the data, like how much information they can tell us between like the gene expression data to the finished data, how much we can learn from the expression to the finished situation. And I'm pretty sure that like basically everyone's pretty familiar with Spearman correlation, and delta score is a very naive but turned out to be very interestingly effective thing that we defined. That is basically the number of helpful genes and its difference with the number of the hurtful genes. And LFC score is a little bit more complicated. We adjust the sign, but basically it's the finished score over a certain threshold, and its product with the log twofold change with the expression score over a certain threshold.
Sorry.
Yep.
How do you get fitness scores again?
What's that?
Get how much the colonies grow?
What's that?
How do you get the fitness score again?
Which fitness score?
Just the nickel fitness score. Yeah.
Oh, the fitness score is what they study in the paper. They basically.
Oh, it's the metric from the.
Yep, that's the heat map. Yep, they basically did a modified z-score using the size of the colony. Yep, and so by doing that, we can like from these 2 vectors drive a anticipation index that is anticipatory or sensitizing, like depending on the sign of it. And that is one condition. In our example, that would be anaerobic versus like 45 degrees from the finished data. But from Nicholas' finished data, they have 324 different stress. So we can expand it column-wise, studying other finished score from the same data set, and then explain it row-wise using like adding more experiment data that we find on GEO. So that will give us a heat map look of the representation of the data. Basically, what we are doing here is a hidden dimension of data analysis. So we have a vector of gene here and a vector of gene here. We then color it using the final correlation anticipation index that we find. And then we build it column-wise, adding more fitness conditions under the same expression conditions. And we build it row-wise, adding more different expression conditions. And if we look at it from this side, it will be a heat map. So that is the conceptual part of like how the data is analyzed. And technically, how is this done? For example, like if we start with RNA-seq data, if you find in Geo, you'll find raw counts and maybe FPKM, depending on how people feel about updating the data. So that is something that we need to deal with. Okay. So we basically first do the gene annotation into B numbers, as we introduced before. E. coli has a well-annotated system of like gene annotation. So we are going to make it as like applicable universally as possible. So we do the gene annotation into B numbers and figure out unannotated entries. And then we figure out the low counts that are possibly noise. And for the raw data, we are going to calculate the FPKM of them. And then we figure out tRNA and RNAs if there is any. And we do a quantile normalization using the bioconductor lemma package. And then we do the replicate filter, filter out genes that, for example, among 3 replicates of the data set, they are of very low reproducibility rate. We are going to filter those guys out. And we collapse the replicate samples of genes into one of each of them. For example, they might have 3 biological replicate for the same stress condition in a same data set, and we are going to collapse them. And finally, we calculate the log-to-fold change of test versus reference. And for the microarray data, like things got a little bit more complicated because data, like there are very many variations of technology that you can actually do microarray. So for expression array, that is the majority of what I'm dealing with now. And we will deal with tiling array a little bit later, but add to our pipeline too. And we can have 1 channel or 2 channel of expression array. And for 2 channel ones, we can have test versus reference. Or sometimes people are cautious enough to do a dye swap. Basically, they swap the test and reference using different dyes. And we need to deal with them separately. And we first bind it into a table from the geo data to a very like RNA-seq count kind of table that we are familiar with. And then we do the same gene annotation and do a signal filter that's analogous to the count filter in RNA-seq, that the purpose is to filter out noise. And then we also do, the quantile normalization of all samples and replicates filter and collapse the replicates. And then we will also have the log-to-fold change of test versus reference as our final output. So, yeah, finally we can enter the matrix. Nope. Yep, here's the matrix. Entering a little bit too fast. And here's a example result from what I have now. So you can see that this is a matrix. This is a heat map. So in the color scale of the color legend is for the normalized anticipation index. The red one means anticipatory, and the blue one means sensitizing. And we have experiment data that is from GEO as the rows, and we have the fitness conditions as the columns. So let's zoom in a little bit to see what is in there and what we can interpret from it. So interestingly, we can see that there are, like, for example, the 5 samples we selected here from GEO dataset. The first one is anaerobic. Apparently, it has a very red box here. And it's actually a stress condition in streptomycin. In this specific case, it makes sense because it's basically saying that anaerobic condition, if you're treating E. coli with anaerobic condition, it can perform better in streptomycin treatment. why it makes sense is that streptomycin requires active transportation into the cells to actually get to actually affect the growth of the cells. And basically, you need ATP to do that, and basically, you need anaerobic conditions to provide enough energy to do that. So in this case, we're very excited to see that it actually makes sense. And we can also see that anaerobic also sensitizing for high pH, which is like more basic kind of environment as what we introduced before in the example. Anaerobic condition can sometimes, well, like many of the times is coupled with low pH environment, so it kind of makes sense too. And in other part of the matrix, there are also other things that we really want to make sense of and study it by follow-up wet lab studies or like more data mining. So that is the matrix. Now we are exiting the matrix. As we say, like there are 4 or 5 different ways that we can score an anticipation index. So one thing that I'm working on with Peter and like would really like your guys' input on is how to select a good anticipation index. So we are thinking about evaluating the way that we score anticipation index using the shift of analog conditions. Yep. So basically what we're picturing is that we define analog conditions as the same stress for expression and fitness. For example, in Nicholas' dataset, it might have anaerobic conditions, and some people on Geo might also have done that study similarly. And we are thinking that most of the time, maybe not always, that cells that are hit by anaerobic condition would be regulating it so that it survives better in anaerobic condition. So we are like basically you are not trying to kill yourself when you're hit by a certain stress, well, most of the time. So we are thinking that if in this logic, you have generally higher anticipation index than the overall anticipation index, which means you will see a rightwards shift of the analog anticipation index over the overall anticipation index. I have been saying anticipation so many times that I cannot pronounce it anymore. Yep, so that's what like a cartoon version of the data would look like. And we would like to study the shift. You can think this is a shift of the peak, so that's basically studying the mode. But after plotting the data, we figure out that it's probably not the best way because like even using statistics is probably not the best way to study if there is a rightward shift. So we add more lines. So we create a stat map of percentile of 5 to 95 percentile of both the overall and anticipatory index, the analog anticipatory index, and see if the analog one is generally better, which means higher than the overall ones. Okay. This is kind of like the output would look like. So the blue one is like, yes, the analog one is performing better, having a higher anticipation index than the overall one. And we can see that, interestingly, mode is actually not doing all that well. And we are looking forward to like narrow down and fine tune our threshold sets to under all this like threshold grids that we have to see if we can find the one that is actually like making us scoring better. So that's quick. Also, the byproduct in this process that is besides building the matrix of gene expression conditions to the fitness, we can also have a matrix of gene condition and the gene expression. Basically, we'll have like we'll have stress as the rows and genes as the columns. We can basically study all the geo datasets that is available and build a huge matrix of like the landscape of its gene expression under certain stress. And we are thinking about doing PCA or non-metric data scaling on it to see if we can find a component or a certain scale of a certain stress is dominating that could probably indicate a certain vector of the genes is dominating the response to a certain stress. For example, if we see a cluster of anaerobic in a corner of it, in a corner of a PCA plot or the non-data matrix scaling plot, we can think that on this axis, there is something about regulating its response in anaerobic conditions. So here is all I got today. It's about 40 minutes, not too bad. Kind of quick, not too quick. And I would like to thank my lab for it, and a special thanks to Marcy that gave me this opportunity to present here. And also, I would like to thank my department. Yep, that's all. I would like to take any questions. Random questions are welcome.
So this one.
Yep.
I think that's well. So your dash line is for analog.
Yep, the dashed line is for analog condition. The solid line is for overall conditions. The color line is specific stress conditions anticipatory index specifically.
There are
Yep.
a very large portion of analog conditions
Yep.
with correlation smaller than 0 on the left side of your plot.
Yep.
So how comes?
We are looking for, we were thinking that there could be a rightward shift. So you can think that a proportion of the overall anticipation index is actually shift this way. So that's why it is here. So it is slightly rightward.
Yeah, so I want to point out an interesting observation, E. coli, though, that's relevant to this. If you take all these nickel stress conditions and you look at the performance of single gene knockouts, you will see that there is a large number of beneficial knockouts under any particular stress condition where you can actually knock a single gene out and the cells do better. So that, because that's to a first approximation like not expressing the gene, it already tells you that the cells are, under many conditions, not giving an optimal gene expression profile for that condition. Because if they were, they just wouldn't be expressing those things at all under that condition. So we know that some of the responses are suboptimal. I think that that's part of what you're seeing here. Probably also our scoring metric is currently suboptimal, and that's what we're trying to fix. But it should be expected that sometimes the cells are behaving the best they could to maximize their fitness under that particular condition. That's probably due to some combination of a couple of factors. First of all, that they haven't been exposed to that condition enough in their evolutionary history to have a really good response to it. Second, there may be bet hedging going on where they're preparing for some other stress condition. Those are really two big ones, I think, actually. Anyway, so you should expect that some of these scores are negative, just because they're not always giving optimal expression profiles.
Oh, I guess I get your question now. Sorry for misunderstanding that. Yeah, and we also do Z transformations to make sure that you see both positive and negative scores, and we adjust the sign so the positive ones are helping and the negative ones are hurting. So the negative scores are the sensitizing scores. Yep.
Just to make your PhD a little harder, would you want to incorporate nonlinear correlations? Because I could imagine there's many nonlinear correlations in gene expression.
That is a really good thought.
Might make scoring a lot more complicated.
Yeah, that would be interesting too. Thanks for making me PhD harder, but that's fun. Anything else? Yep.
You had spoken about PCA.
Yep.
Are you trying to use that to identify underlying stress response that you find irrespective of what stress? Is that what the point was?
Yep, you got it. I have tried doing PCA on that, but the current difficulty is that you can understand that every data set is not studying the same set of genes, although they are on microarray. So I find out that PCA plot is highly confounding with the data set that we are finding it so far, because we are not categorizing by the biological differences. We are more categorizing by the technical differences of like, for example, they are studying the first 100 genes or the last 100 genes. So they have similar missing genes, they got clustered together. And that is the biggest difficulty we have been looking at. So that's why we are trying to do non-metric data scaling that's not so sensitive to missing genes. But like, given good normalization, and we also have been thinking about like using lasso or like confidence interval kind of method to permutate the genes that we are not observing, the missing genes. So that will also add to our power to do PCA on it and figure out the pathways. Yep.
These groups go back to like some reference culture when they start all these experiments, or are they using things inside the lab that have been passed through 10 generations of graduate students?
These are almost always very closely descended from the sequenced culture. So there is one stock center that almost everybody gets their stuff from. They're getting it pretty fresh. So you know it's
you guys all start out from that.
We all start out from that. So you're no more than a couple overnights away from that culture in most cases. Now, overnight cultures. Now, there are, for example, a few variations that have shown up across the different copies of, say, MG1655, but because we can sequence them so easily, we know that like there are 7 mutations between like the 2 extremes that we've seen in the variations of MG1655 that people have. But that's not too bad. We know what they are. We know to watch for those. But in general, they're very, very closely related.
And all the labs doing this kind of work adhere to that.
Yes, they do. It is considered horrible practice, for example, to perpetuate a culture the way you would if you were doing mammalian cell culture or something like that, because you would accumulate a lot of mutations. So the first thing you do when you get strain, you grow it once and you freeze more than you need in 10 lifetimes. So you're always going back to that exact same stock culture. And so the entire community is very careful to maintain that homogeneity as much as possible in cultures. Which is part of what makes this a good system to work with, I think, and why it's actually possible to consider doing these kinds of comparisons.
So that's it.
Have you tried to cluster the conditions and see whether your cluster makes sense?
Yes. Like, you mean the experiment conditions, right?
So you have your last but one slide where you have
Oh.
matrix to this condition.
That one? Oh.
Yeah.
This one?
So if you do hierarchical clustering of conditions, would they make sense?
Basically, that would be the equivalent of clustering the heat map, right?
Maybe, I'm not sure.
Yeah. If you're talking about clustering the conditions that we have seen in the heat map, some of them make sense. That, for example, like different anaerobic conditions got clustered together, but like some of them is still like due to our scoring methods power in like analyzing missing data. Some of them is still highly confounding with the data set that they are from. So we might see like 10 samples from the same data set clustered together, why many of them like is clustered by the stress itself. So that's one thing we need to improve in our anticipation indexes to deal with all those missing data. Yep.
The anaerobic and streptomycin,
Yep.
you saw that anticipatory effect, and that made sense. Did you see any results that just you're like, that doesn't make sense at all? Why would this be?
Yeah. Like, if you have to say the amoxicillin and oxacillin wouldn't make sense because they are both penicillin, you would expect if you treat it with amoxicillin, it will start dealing with amoxicillin right away. So you're basically dealing with a wide spectrum penicillin. But then you treat it with oxacillin, which is a narrow spectrum penicillin. You kind of expect that now that they are ready to start to deal with penicillin, they probably won't do so bad. But this case is actually sensitizing. So this is an interesting thing.
So we want to do the experiments and find out.
Yep. Yep.
So are you actually going back to experiments, or that's not part of your?
I have been touching bench.
You can think about it.
Yeah, I have been like doing a little bit of wet lab, but most of my work now is still on the dry bench.
Thank you.
That's all. Thank you, guys.